Hi, everyone, and thank you for coming today. So the two sessions that we are going to have today are going to be slightly different. And they're going to be slightly different because what you're going to see is state-of-the-art research. In fact, this first none of the two presentations I'm going to give have been published yet, so it's kind of slightly confidential. But I think it's good that you guys have a little bit of a taste of what it is to feel where the frontiers of research is in genetics, genomics, and hopefully you'll get a little bit of a bit of excitement with respect of the amazing things that science can do to help us understand very important questions. So this first presentation that I'm going to give, the long title is what Ekaterini put on the email. So we're going to look at the clinical interpretation of the genomes of descendants of the Inca Empire. So the Inca Empire, whose capital was in Cusco, Peru, is a very rich cultural heritage of more than 12,000 years since it was first colonized from actually people coming from Asia, from the Bering Strait. They went all the way from Alaska down from the coast to South America, and they established themselves there around 12,000 years ago. And what we see today in Peru and around South America is a lot of admixture, a lot of mixing between different types of populations. And if we want to really understand the genetics of Native Americans, it's really important that we are able to obviously run all of the different algorithms that are available to see what kind of data, what kind of variants they have, what kind of mutations they have, because most, and this is going to be with regards to the second topic I'm going to be presenting today, most of the data that we have as of today is European based. And that imposes a tremendous bias in our ability to apply that knowledge into people who are not of European origin. And as part of the sort of, it's not just a question of justice, that everyone should have access to the benefits of the science that comes from the human genome, but also it's a question that everyone benefits from having these diverse populations understanding. Because if we're able to see how the different diverse populations react to specific illnesses, that can help us then see for the better studied populations how those illnesses actually are relevant. What things that are relevant for different populations can also be relevant to the better characterized populations. And the Indigenous Americans are among the least represented ancestries in global reference resources. So there are around 50 million people who could be classified Indigenous American, and less than 0.1% of all of the data sets that we have in reference databases from which we then derive and are able to interpret clinically the consequences for those patients have data there. So the fact that there is this extreme underrepresentation makes it difficult for these populations to benefit from the latest advances. And so because in fact, the analysis that I'm going to present today here, these populations have never been studied before. So in a way, it's a little bit like a conquest, so to speak. We're in an exploration journey into trying to understand things that have never been seen before. And being able to understand the genomes of descendants of the Inca Empire, who are people living in today modern Peru, has allowed us to really reconstruct history. And in a previous study with fewer people, it was found that no later than 23,000 years ago, the Native American ancestral population diverged from East Asia and then came down into the Americas and entered into a period of isolation. So we know that the first settlers, and this is just looking at genetic data, okay, from these individuals, we think that the first settlers arrived around 12,000 years ago, and then there were three branches that continued with the colonization of the land. So there is the Amazon branch, the branch that goes through the coast, and the branch that goes through the Andes. And these are very different environments because the coast is one of the driest deserts that you can think of, and there's very little water. So people who actually went through the coast would go from river valley to river valley. So rather than going through the actual line of the coast, they would go up through the valleys, which would have a lot of water from the rivers coming from the Andes, and then through the rivers, then they would cross via the mountains down into the coast again. And so those patterns have been really seen here, because what we see, and this is not very well displayed in this graph, what we see is that actually most of the colonization of those lands by these ancestral people was mainly through the mountains. And then the people that ended up in the Amazon became adapted to the things of the Amazon, the jungle, all of that environment. Whereas the ones that stayed in the mountains, then they became adapted to high altitude. They had genetic adaptations that allowed them to survive in those conditions. And the same for the coast. And by looking at their genome data, we actually were able to see that in 1532, when the Spanish conquered the South America, it was at the top of the Inca Empire. We're talking of Cusco, somewhere here being the capital. So as soon as the Spanish came, they brought with them all of the diseases that the Eurasian had been adapted for, and they brought tuberculosis, influenza, herpes virus, smallpox, and the viruses were spreading faster than the Spanish, and around 90% of the population died around those decades because they had no exposure to those particular agents, and it meant that they weren't able to cope with that. So in fact, that obviously had a tremendous genetic component, which I'm trying at the moment to identify. So can we actually find those susceptibilities that we found in those populations actually reflected in their genome? And I have already some indications, which I'll show you in a moment, that we already begin to see some signatures of specific genetic mutations affecting genes involved in immune response, as well as things related to adaptation to infections. So then there was the very interesting thing that suddenly you get Europeans coming in to Latin America. Obviously, the whole system, the whole civilization was completely altered, and we had that the Spanish actually mixed with the mestizos, with the Amazons, with the people in the coast and the Andes. But something quite interesting was the fact that that mixing actually didn't happen specifically, and this is something that we were able to, that the previous study with this data revealed, was that the mixing of the Europeans with the natives actually happened not when the Spanish came, but when Peru became independent from Spain around 1800s. And at that point, there were wars, and again the whole social classes were halted, and at that point we can see in the signatures that is when the mixing of the Europeans who were already living there for a couple of centuries actually became really mixed. And so what you see today in modern Peru is that there's a huge amount of mixing with the Europeans. There were also some people from Africa who were brought as slaves, and then they also stayed there, and they now also mixed. So you find Afro descendants who have a little bit of Spanish and a little bit of Native American. So it's a very complex, yet fascinating, yet under-discovered case. And what you find is that in places such as Lima, which is the melting pot, Lima is the capital, it's the melting pot of Peru, you're going to find the greatest degree of mixtures between the different types of populations. And then the further you go from the coast and Lima, you're going to see that there is less and less mixing of these peoples with other populations. Many of them have been isolated for a long time, and we had the opportunity to have genome data coming from some of those populations, and we can see, for instance, some very important susceptibilities to disease that those isolated populations don't have when they come more into the sort of more civilized, more sort of mixed areas of the country. So I'm going to give you some initial preliminary explanation of the first type of analysis that we developed. So the first analysis was with 150 whole genomes. So you probably remember what whole genomes are. So you basically take the DNA from the patient, you cut it into millions of fragments, then you run those fragments through a sequencing machine, and then you try to recompose those fragments back into the actual structure of their chromosomal assemblies. And based on that, then we can compare the differences that we observed in the individuals with the reference map of the human genome. And those bits that we find different, we use them for further analysis. So for those 150 whole genomes that we analyzed, we found 13 million single nucleotide variants, so 13 million SNPs. you may see here that it says biallelic. Some SNPs have more than 2 alleles. You remember what an allele is? So we selected only those SNPs that had 2 alleles for the purposes of much ease in terms of them further analysis. We also ran, so that's our starting point for those 150 genomes. As you can see, you get a huge amount of data, right? 13 million data points. Then we ran something called kinship coefficient, whereby you take those 13 million data points, and then you basically prune those that are too similar, right? So running a particular program, I think it's called King, you can actually, when you consider the population, you can identify those that are first to third degree relative. So first degree relative would be your sister, your mom, people who are directly related to you. Second would be, for instance, your uncle, your grandfather, your grandmother. Third would be your second cousin, something like that. Okay, so if we have populations that have 2 close relatives, that is a little bit of a problem because it biases the results. And so you want to have people who are from different families, because that way you have a much better representation of the real diversity happening in those populations. The human genome reference that we used is called HG19. I think I might have mentioned it earlier. So HG19 is the CRGA, it's also called CRGA37, and CRGA37 is the 37th version of our human genome draft reference. I'm not going to go into this more detail, but just simply to say that those single nucleotide variants that were of low quality, we removed. So in the end, we ended up, as I said, with 13 million variants after a long process of pruning, filtering, quality control. And here you have the first populations, what they look like. And I have ordered them according to their most perceived, most native to least native. So in this case, we have the Matzes, who are completely in the Amazon. They've been isolated for many generations. Then the second is Uros. So these Uros ones, we took them from a lake called Titicaca Lake. Not sure if you heard of it, but this lake is around 3,500 meters high, and it's a complete rarity, right, on the top of the Andes. So these populations, the Uros, which are this second one, this lady is a Matze, this person is Uros. So they live in an island inside the Titicaca Lake, and basically, again, a degree of isolation, which is very difficult to find nowadays, and most of all, to have the opportunity to get the DNA from these people, so that we can analyze them. Then we also have the Chopccas. You can see them there. The Chopccas are well known in Peru for their warring spirit. So they have a reputation for being the ones that become soldiers, that they're apparently very good at fighting. And then we also have the Moches. You can see the Moches. Then Iquitos, you see this person here, and then Cusco. So as you can see, there's a huge amount of diversity in the different populations, and as I said, these populations have never been characterized before. So the first thing that we did once we took those data sets, we use from Ensemble. You might have heard of Ensemble. So we used a tool called variant effects predictor that basically accepts the data that you give them. Obviously, it has to be genome sequencing data in the right assembly. So in this case, 37. We upload the data, and then it gives us information about each of those 13 million variants, and it basically tells us information about whether they have been seen in other populations, whether they are inside genes, and the effect that these variants could have in the genes in terms of the function. Are they part of an intron? Are they part of transcriptional regulation site? And so on. So from the 150 whole genomes, we took out those those variants. Sorry, did I say genomes? So from the 150 whole genomes, we got 6 million plus variants that are either non-existent or less than 1% frequent in any of the existing global population reference datasets. So they are all rare, right? You can see, just to get started, you can see that almost half of all of the variants that we have are very, very rare. So they haven't been seen before, right? So the variant effect predictor allows us to exclude common variants, where we filter out those variants that are co-localized with an existing variant that has a frequency greater than 0.01 in global populations. And then it allows us to apply further filters according to the potential effect that any of these variants could have in terms of the disruption of genes, for instance. So here you can see the interface that the variant effect predictor provides through Ensemble. There's a number of options that you can select, but here I'm just showing that this was the advanced filtering that I decided to use for this particular characterization of variants. The thing with this type of analysis is that there are so many data points that in the end, your whole purpose for applying this filtering is to end up with some kind of manageable amount of data points where you can begin to do and make some initial study, because so you really want to filter out as much as possible in terms of potential relevance to functional effects. So we applied the filtering criteria that from those 6 million variants that we only wanted to see those variants that were protein coding. I lied in regions of the genome that are involved in that are coding that encode for a gene. But not just that, because there are different types of variants that you can have in a gene, right? So you can have synonymous variants, you can have substitutions, you can have inversions, you can have deletions. So we only wanted to have those variants within genes, within coding regions, not introns, for instance, that have a high impact. So what are the variants that have high impacts? For instance, we have things like frame shift variant. Do you know what a frame shift is? It breaks the codon, right? And so, yeah, it shifts the reading frame. Stop lost. Yeah, exactly. So the stop codon disappears, and then it keeps on translating the protein, right? And then stop gain. you gain a stop codon where there shouldn't be one, right? So those are examples of the high impact type of mutations that you can have in a gene. So when we look at the numbers now and try to classify them according to these criteria, I told you that we started with 13 million SNPs. We reduced them to 6.2 million with the filtering criteria that they should be rare or not characterized as of today. And then from these so we had 2.8 protein coding variants, and from there only 1,235 were high impact. So most of them stop gains, some of them splice donor, some of them splice acceptor. So these splice, can you think of what would be the consequence of this?
So which splice?
Yeah, so we have some mutations that cause or affected the splice sites. Yeah, the splice donor or the splice acceptor. So the splice sites are where you have the limit between an intron and an exon. So if you mutate any of those splice sites, then you may end up retaining the intron instead of splicing it out, for instance. So that's the type of consequences that the splice mutations have. And then start lost or stop lost. The next thing that we wanted to do is to see from those high impact mutations, is there anything that has been known or that has been found or annotated that we know about those variants, right? There's a little bit. So we found that 22 of them have been already characterized as pathogenic or likely pathogenic, certain significance and so on. But the vast majority of those 1,200 are unknown. In other words, we have no idea. So we know that they have a high impact in the gene, but we have no idea what kind of functional consequence. Are they pathogenic? Are they not pathogenic? And so you can see here the actual variants. So in this table, what you see so those 1,200 variants, I've prioritized them according to their frequency in populations, right? We said we had 300, we said we had 150 genomes, right? So each genome, each individual has 2 alleles, right? That's why we have, here what we are doing is counting alleles for the variant that is causing a high impact, okay? So we have a total of 12 Matzes, which are the most indigenous of all of them. We have 30 individuals who are Uros, 30 who are Chopccas, 30 Moches, 16 Iquitos, 16 Cusco, and 16 Trujillo. So, but you can see that for that particular mutation, which is a splice donor variant, all of them have both alleles with this high impact mutation affecting them, right? So we are prioritizing according to their frequency in the different populations, all of those high impact variants, right? And so what we can see is that I've added a little bit of some highlight here, but basically you can see those gene symbols. So those are the gene names associated to those particular variants. So the gene symbols that you see in red, bolded red, those are genes involved in immune function or immune response. Okay, so we see when we then look at the potential function that we have for the genes that could be affected, we see that there is an enrichment to susceptibility to infectious disease and an enrichment of highly mutated genes which are very well present in those 150 individuals, which are very rare in the rest of the world, but somehow those genes seem to be involved in immune response. Okay? Any questions so far? Can you see the kind of research, what research is like? Right. Okay. So those same genes, now I'm representing them according to the normalized frequency. So basically, the first three genes, all of them are seeing all of their alleles mutated in all of the populations. But then we see that there are some other genes whereby they're highly mutated in some populations and some of them are not. So here I'm plotting the frequencies, and you can see that for this Matzes population, they have a high rate of mutation for this particular gene, which is the MYL12B, then another one here, and another one here. And you can see those genes, what potential function they are involved, they are related to. Again, you can see a slightly overrepresentation of genes that are involved in immune response and some other functions. For the second population, the Uros, who are the ones that live in the Titicaca Lake, you can see some genes that are more frequently mutated in those populations. And you can see those genes, potentially, what function they would be involved in. Okay? So, next, we also had a 722 arrays. You've heard of what a microarray is? Have you heard of a microarray? Anyone doesn't know what a microarray is? Anyone would like me to explain what a microarray is? So a microarray just is a chip it's a glass chip that has hundreds of thousands of wells, and each of those wells have a particular probe that measures, in this case, the presence or absence of a particular SNP. So it's much cheaper than sequencing, but it only allows you to discover the presence or absence of mutations that have already been characterized. So there are pros and cons. So in this case, we had 722 arrays for all these populations. So we have the descendants from Africans and the same populations as before, and many others. In total, we have 25 different populations, right? So the thing that we did with this much greater number of individuals, because instead of having 150, now we have 722. And these 722, when we then try to look at how the variants that we observe for those individuals, we decided to test them in pharmacogenes. Yeah, so we know that drug responses are affected by your genome. We know that when you take a particular medication, that medicine is going to be metabolized in your gut into its active component or active principle, which then will have its function. But that metabolization is conditioned by the sequence of the gene that encodes those particular enzymes. So we find that there are enzymes with different variants in different populations that affect their ability to metabolize drugs. For the better, for the worse. Obviously, what you want to have is to be able to predict the exact metabolization status of one particular patient for a drug. If that patient, you give them, let's assume that patient comes from Africa, and then you give them the dosage of that drug based on what we know about Europeans, chances are that some of them will have adverse drug reactions because they might have different variants that make them, for instance, ultra low or poor metabolizers or ultra high metabolizers, and so the dosing algorithms, which are based on Europeans, will have an effect for underrepresented populations when they then have the same dosage as the Europeans. So with this premise, there is a database called PharmGKB, whereby it contains all of the gene-drug reactions that have been observed to date. So it has the collection of all known pharmacogenes, and then the different variants that have been characterized as of today for all of those genes, and the potential implications in terms of the different alleles that people have based on those variants, and that obviously has a consequence in the dosing that you want to have for those patients. So based on that, we found that out of those 722, all of them had unfavorable response for the pharmacogene F5, which is involved in anticoagulation. And we also found another interferon L3 gene, where we find that a huge amount of the vast majority have unfavorable response. These genes actually are produced in response to viral infection and can elicit antiviral state as well as modulate adaptive immune responses. And they are mostly associated with clearance of hepatitis C virus. So something that hasn't been observed before, but basically we found that these populations are going to be liable to have susceptibility for that particular gene when they have antivirals for hepatitis C. So for this study, then, the conclusions that we find so far, and we've just scratched the surface here. There's a lot more that we are going to do in the next few months. We find clear indications that genes are involved in immunity and infectious disease susceptibility. What I haven't been able to find yet is whether there is a direct relationship, which I think there is, but I need to have further evidence, which I'm going to try and explore further, of the relationship of some of these susceptibilities to the diseases that affected the Inca Empire descendants, or the Inca Empire people when the Spanish went in the 1500s. I think it would be a great historical finding to be able to see that actually, we do see precisely those things that were reported in the historical documents, that actually, yes, that we see its reflection in the genomes of the direct descendants from those populations. There are some differences between ancestries, and that may explain that some populations, such as Uros, they have a much greater susceptibility to tuberculosis than some others. And we have seen that there are clear implications for pharmacogenomics dosing that should be taken into account if these underrepresented populations are going to benefit from the advances that have happened in the past couple of decades as a consequence of the Human Genome Project. And that concludes my first presentation. I'm not sure if you would like to ask any questions. Would you like to have a little break? Cool. Little break then.